thank you everyone for attending the last climate seminar of the year. I'm standing in for Dr. Ken, as you can tell. I'm excited to be hosting Dr. Jennifer Head. Dr. Head is an assistant professor in the epidemiology department and a member of the Institute for Global Change Biology, both at the University of Michigan. Her research applies mathematical and statistical models to understand the impact of climate variation, environmental disturbances, and zoonotic hosts on infectious disease dynamics. A major focus of her work is on the emergence of environmentally acquired fungal infections such as valley fever. Her work combines methods from epidemiology, data science, econometrics, environmental health, and ecology, and she collaborates closely with state and federal agencies to help convey findings into actionable public health outcomes. So hopefully you can help me welcome Dr. Head.
Well, thank you so much for that kind introduction, and thanks to Dr. Turian for the invitation. I'm really excited to be here today and meet all of you. And I think it's really phenomenal that you have this Center for Climate Change and Health. It's such a pressing problem. So I think it's really great that you're all are really on it and topic. I wanted to start out with just a disclaimer that this work is done in collaboration with the Department of Public Health and EPA, but does not necessarily represent the and I also want to start out with just my acknowledgement slides just to demonstrate that the work that we're talking about today really is the work of a lot of individuals spanning multiple institutions. And so when I say we in this presentation, I'm referring to a giant body of scholars. So maybe many of us in this room already think about this a lot, but a recent study has shown that over half of known by climate change. And one class of pathogenic disease that I think doesn't get a lot of attention, but which I think about a lot, is fungal pathogens. And they are emerging globally. So in 2022, the World Health Organization came out with the first ever list of priority fungal pathogens, which was in response to evidence that fungal pathogens were potentially adapting to warmer temperatures. We're recently in this national climate assessment shown on the bottom left here. There was this map showing emerging climate sensitive infectious diseases, and fungal pathogens were also represented very clearly. So fungal pathogens are a particular concern because there are no fungal vaccines at the moment, and there is rising antifungal resistance. So the fungal pathogen that I have been thinking about a lot over the past 5 or 6 years is, or the disease that it causes, known as coccidioides, which is difficult to say and even harder to spell. And so we often call it valley fever or cocci. And maybe could I have a show of hands who in this room has ever heard of a disease called coccidioides or valley fever? Okay, so actually quite a lot of individuals. So that's fantastic. When I moved to California, I had never heard of this pathogen. But coccidioides is a disease caused from the Coccidioides fungus, which grows in arid soils. And the endemic area is much of the southwestern US, but particularly California and Arizona, where over 95% of cases are reported. So I have focused primarily on coccidioides in California. And as you can see here, this area kind of outlined in the darker black is the Central Valley of California, and that's where most of cases of valley fever are reported. Particularly that county in dark red is Kern County in the southern San Joaquin Valley, where historically the incidence has been the highest. But we have seen rapid increase and expansion in incidence rates over the past 2 decades. So in Kern County, in the dark red, we've seen incidence increase over 8-fold in the past 2 decades. But even more concerning, in some of these coastal regions in northern San Joaquin Valley counties, we've seen incidence rates increase by over 15-fold for the past 2 decades. And there's a lot of speculation that climate could be playing a key role in this process. And so if we think about the life cycle of the Coccidioides fungus in soils, we can start to conceptualize where climate might play a role. So Coccidioides grows in the soil in these long branching chains that are called mycelia. And when the fungus has access to nutrients and moisture, it can grow. But when the soil gets arid and nutrients are limited, those branching chains get brittle and they fragment, releasing individual arthrochonidia that can then become carried in fugitive dust emissions and inhaled by a susceptible host, causing pretty severe respiratory disease in some cases, and even progressing to chronic infection in about 5% of individuals. So because of that process of the wind coming along the spores, we suspect that airborne spores can be carried in fugitive dust emissions. And so people who work in dusty regions or outdoors are particularly at risk, including construction workers. And we also see disparities in both incidence rates and mortality rates that may be driven in part by disparate exposure to these environmental hazards. So we do see higher incidence rates among particularly outdoor workers, including construction workers. There have been outbreaks among agricultural workers and wildland firefighters. We see really high incidence rates among people who are incarcerated, and African American and Hispanic individuals have higher incidence and mortality of disease. So my goal that I'll talk about today is to identify environmental factors affecting the emergence of valley fever in California. And so in this presentation, I'll go through kind of three main buckets. The first looking at drought and precipitation volatility in the state. The second considering dust emissions, looking specifically at wildfire. And then the third talking about some field research that we're doing to try to understand where Coccidioides lives in the environment and its association with other animal hosts in the area. So I'll just jump right into drought. So the research question that we partnered with the California Department of Public Health to ask is, what is the causal role of precipitation, temperature, and drought on the emergence of valley fever in California? And this work has been published, so you're welcome to check out. So specifically, we wanted to tease out sequences of temperature and precipitation, knowing that this fungus has a long life cycle. And so the order of events in which precipitation and temperature are experienced can matter. And that's what I'll talk about first. So like I said, we partnered with the California Department of Public Health to gain access to all the reported surveillance cases on valley fever in the state since 2000. Then we geocoded these and aggregated them to the census tract level. And linked information on census tract level cases to lagged environmental data on temperature, precipitation, other environmental covariants like soil texture, elevation, and then spatiotemporal variables. And we lagged temperature and precipitation by up to 36 months with the understanding that the growth process of this fungus in the soil can take a long time, and a lot of the upstream factors can influence growth rates. So then for each county in California, we regressed cases against lag temperature and precipitation using distributed lag nonlinear models. And so that enabled us to, for each lag of temperature and precipitation, arrive at some nonlinear exposure response relationship between our environmental covariate of interest and cases. And here's just an example of such a nonlinear relationship shown here. So we repeated this for each county, and then we pooled the counties using a meta-analytic approach. So then we computed the incidence rate ratio for an IQR increase of our environmental exposure of interest. So what you're seeing here, if I can use this now. So this is just an example here where the 25th percentile of monthly summer precipitation is at 0. The 75th percentile of monthly summer precipitation is over here at about 2. The incidence rate ratio associated with that increase is 0.95. And so we plotted that on this graph over here. So what you're seeing on this graph, the y-axis is the incidence rate ratio of valley fever per IQR increase. And then on the x-axis is the lag from disease onset to when precipitation or temperature was experienced. And we modeled just cases that occurred in the fall for 2 reasons. 1, that is when most cases of valley fever occur in California. And 2, doing so enabled us to map those lags onto specific times of year, so we can back out, whether a wet winter or a dry summer is what's driving this. Okay, so I'm going to walk through what we found, and on the upper right-hand side of this slide, I've also included a diagram of the life cycle of Coccidioides, so we can also start to conceptualize how the lags are mapping onto parts of that life cycle. And so anything, I'm doing this for precipitation here, so anything IRRs above 1 means that higher rain is associated with more cases, and IRRs less than 1 mean that higher rain is associated with fewer cases. So what we saw is that in the short term, higher precipitation is associated with fewer cases. And this fits in with our understanding of kind of what's happening before disease onset and which dryness could be associated with fugitive dust emissions. In the 5 to 9 months prior to disease onset, we see that higher precipitation is associated with more cases. And this fits in with when we think Coccidioides might be growing in the soil and using all that moisture and nutrients. And this also kind of maps onto winter in California. And then what we saw in the 1 to 3 years prior to disease onset is that as a main effect, the environmental conditions do not have that interesting of a story, but they were important effect modifiers of what was happening in the winter. And in particular, we saw that if there were dry conditions in those 2 to 3 years prior to disease onset, it amplified that positive effect of winter precipitation. And we'll talk a little bit more about that and why that might be to come. So then we also wanted to look at how this relationship varied across the counties in California. So we looked particularly at the times of the year when the environmental variables were having the strongest effect. And this was winter precipitation and summer heat. So each of these lines here is representing a county in California where the exposure response relationship is colored by their either median winter precipitation on the left or median summer precipitation on the right. And what we found is that hot, dry counties are strongly influenced by precipitation. So on the left-hand side there, you're seeing exposure response relationships between precipitation and valley fever. And you can see that increases in precipitation in those counties have strong positive IRRs to a point. I'll note that precipitation in those regions rarely passes that hump. So usually precipitation is associated with increases in those dry counties. But in these hot, dry counties, the relationship between temperature and incidence is not as strong. In contrast, if we look at the cooler and wetter counties, we see that the relationship with precipitation and cases is null or even negative, but there is a very strong positive influence of heat. And so what we speculate might be happening here is that the fungus requires 2 things to spread. The first is moisture and the second is heat. And in those cooler, wetter counties, maybe they always get enough precipitation, but what they don't always get enough is heat. Whereas in the hotter, drier counties, they're always hot and dry in the summer, but maybe what they lack is that precipitation in the winter in order to grow. So once we kind of teased apart some of the sequences of precipitation and temperature and their importance, we wanted to dig a little bit deeper into the effect of major droughts on regional incidents in California. Knowing that California has had some pretty severe droughts in the past few decades, the most notable ones being between 2007 and 2009 and 2012 and 2015. And so we did so using an ensemble prediction algorithm approach. So first, we came up with a variety of candidate algorithms that ranged from some pretty simple generalized linear models to some more complex models and some random forest models. We then trained these models on the observed data using leave-out 1-year past validation in order to determine their out-of-sample prediction error. And then we computed a weighted average of these models in such a way as to minimize this out-of-sample prediction error. So what I'm showing here is a map of Kern County. So we did this for each county. What is shown here in the black is the observed cases, and then the black line is the model prediction. So in general, we were able to achieve a pretty good prediction to our observed data. Our R-squares were above 0.9 for all the counties. And then in gray, what you're seeing here is the 2 periods of drought that we wanted to test. So then we used a G-computation approach to estimate how much incidence would have occurred under this counterfactual scenario where the 2007 to 2009 drought didn't exist or the 2012 to 2015 drought didn't exist. So during those drought periods, we deterministically intervened to set values of rainfall or temperature that were rainfall less than the mean or temperature higher than the mean back to the mean value in that census tract. And then we plugged that into our ensemble model in order to generate predictions of what we would have seen in California had those droughts not occurred. And then we subtracted the observed from the counterfactual predictions to arrive at an estimate of drought attributable cases. And so what you're seeing here in the colored lines are those no drought counterfactual scenarios. So the blue line here is the no 2007 to 2009 drought. The pink is the no 2012 to 2015 drought. And a few patterns that you can see is that during the drought period, we observe fewer cases than we would have expected had the drought not occurred. But if you look at the 2 years following drought, we actually observe more cases than we would have expected had the drought not occurred. And so again, similar to what we saw before with the 2 to 3 years prior to disease onset being effect modifiers for the wet winter, we're seeing that something about drought then makes wetter winters support Cocci transmission a little bit more. So if you subtract the two cases, the counterfactual from the observed, again, we're seeing that there was a net decrease during the droughts in valley fever cases that is attributable to the drought, but then in the 2 years following the drought, we see an increase above what is expected, and this net increase more than offsets what the decline during drought. If we map the changes due to drought onto the study region of interest, we see that in almost all the counties, there was a net increase in valley fever cases attributable to drought, except for in Kern County, which is the driest and hottest county that historically has had the most cases of valley fever. And in particular, we see positive effects of drought in these more coastal counties and the northern San Joaquin counties that have been experiencing disproportionate increases in incidence. So I will talk a little bit more about why we think the relationship between drought and valley fever is happening, but some hypotheses is that during drought, Coccidioides is a pretty hardy spore and it can live in the soil, but its microbial competitors do not. And so during drought, the microbial competitors die off. When the rains do return, Coccidioides can grow uninhibited by other competition. Another hypothesis relates to the die-off of rodents during drought. And I'll talk a little bit more about that later on in the presentation. So just to summarize a little bit of what we've seen here, we found that drought first displaces and then amplifies incidence. We see that the amplification due to drought is strongest among these wetter and cooler counties. And in these counties, incidence is also especially sensitive to summer heat. And we know that drought frequency and severity is expected to increase in the US, along with this precipitation volatility, by which I mean swings between extreme drought and extreme precipitation in the form of largely atmospheric rivers. So I want to demonstrate what has kind of happened in recent years because this study sort of stopped or the analysis of this data stopped in 2020. But between 2020 and 2022, California experienced another extreme drought such that there was a great paper released saying that between 2020 and 2021 was the driest 22-year period since 800 CE. And you can see that this is a picture from our field site in the Central Valley at the end of that drought in springtime. But then atmospheric rivers came at the end of 2022 and the landscape looks quite different due to that. And so because there was that drought that ended in 2020, followed by atmospheric rivers, we anticipated that 2023 would be a really big year for valley fever. And so we worked with state health in order to put out these warning messages, just letting the public know and alerting physicians that we expected a lot of valley fever cases. And indeed, 2023 was the highest year on record in California. Atmospheric rivers have continued in California through 2023. And so we are already anticipating that 2024 will be a very high year for valley fever. So with the ensemble model that I just presented, we're working on building a 2024 forecast that will hopefully release quite shortly, but already anticipating maybe a little bit fewer cases, but almost as many cases as was seen in 2023. Okay, so I am now going to transition a little bit more to some other major climate challenges that California is facing and their potential role in the emergence of valley fever in the state. And in particular, the one that I want to talk about is wildfire. So as with drought, the frequency and intensity of wildfire is expected to increase in the Western US. And this could have influences on valley fever incidence for a few reasons. So first, what we're seeing here on the left is just a diagram of a landscape prior to fire, where maybe we have this diverse soil community. And then during fire, there is massive mobilization of soil. So spores could be carried upward in the wind and dispersed. But on the other hand, spores could be inactivated by heat. So from this, we could hypothesize either increases or decreases in valley fever transmission. And then in the longer term, we know that after fire, there is increased wind erosion of soils, which could increase valley fever transmission. On the other hand, it could sterilize soil communities, which could be good or bad for Coccidioides, or it could deplete nutrients, which might be not so helpful for Coccidioides to grow. So we first tackled this question using sort of data-driven approach where we asked what are the immediate and delayed effects of wildfire on valley fever incidence among people who are living near wildfire. So to answer this question, I worked with Sophie Phillips, who is now a doctoral student at UCLA and is a really fantastic individual. So we decided to use a synthetic control approach. And so our first step here was to identify our treated units and the synthetic control pool, the eligible control pool for each fire exposed region. So we downloaded a map of fire burn areas. We subsetted to those that were just larger than 2,000 acres. And we drew a hexagon around it to be the treated units. And then we identified as the eligible control pool other hexagonal areas that did not have spatial overlap with the exposed unit and did not have a fire that occurred during its in the 3 years before or after the ignition date of the fire of interest. And so that resulted in about 26 to 34 potential eligible control units for each of our treated units. So then we computed the synthetic control by matching on population, mean daily temperature, total precipitation, vegetation coverage, wind velocity, and pre-fire case count. And so what you're seeing here on this graph is an example of our pooled effect. So in the black line is the observed valley fever incidence rate, and the dotted line is the synthetic control, the incidence rate and the synthetic control. We fit it on the 3 years prior to fire ignition, and then we're going to look at what happens in the 3 years after. And in that shaded region there is the 95% confidence interval. So then using the synthetic control, we estimated the number of cases in the post period that would have occurred under this counterfactual no fire scenario. And so again, what you're seeing here is that the observed valley fever incidence rates are falling within that 95% confidence interval on our synthetic control, which suggests that there was not a significant association between fires and valley fever incidence rates in those who are living around the fire, at least on average. So indeed, the fire was associated with an 8.6% increase in cases, but this was not significant at the 95% confidence level. So we did look at the effect of each of the 19 fires that ultimately met our inclusion criteria individually and pooled. And so while the pooled effect was non-significant, we did identify 3 fires that did have significant positive effects in at least 1 point in the 3-year period following the fire, either the full 3 years, the first year, the second year, or the third year. And these 3 fires had a few things in common as compared to the other fires. One is that they had higher transmission pre-fire than some of the other fires, suggesting that valley fever was or Coccidioides was potentially more common in the soils there. They were near areas with higher population density, which might suggest that we just had greater statistical power. And then they were also larger fires relatively, so they were all larger than 5,000 acres. So I think this study had a lot of limitations, notably being the ability to pick up a statistical effect between fires. Also, usually where wildfires occur in California, it's sort of at the periphery of the Central Valley. So it's not really in the regions where either a lot of people are living or where we think valley fever or Coccidioides is most likely to be in the soil. And so what we're excited about doing next is maybe starting to combine some of this epidemiological evidence with field measurements in order to better help us understand Coccidioides aerosolization and transport. And so we're interested in thinking about fire, but also other sources of dust emission in the state, including construction, the digging of oil and gas wells, which is a very dust generating process, and also just fugitive dust emissions through wind. And so we've partnered with the EPA in order to understand how these remotely piloted air filtration methods might be uniquely situated to help us understand these questions. So these are our drones, which you can see here on the right-hand side. And they're quite flexible because they can access various heights in the air column. You can maneuver them to different locations, and they record the GPS coordinates, so they're quite spatially precise. And others have worked, some of our collaborators have flown these drones through wildfire smoke and have detected various microbes in the air associated with fires. So we wanted to know, can this method work for Coccidioides, because it's not previously been done, and the limit of detection for spores and airs might be pretty low. And indeed, if we think about past air sampling methods for Coccidioides, really the only methods that have been successful in detecting Coccidioides in air have been these sort of large air filtration units that are sampling maybe tens of thousands of liters of air at a time. There have been some successful efforts with passive means of collecting dust, but it's quite limited at this time. And so we just wanted to conduct a pilot study to ask, can we detect Coccidioides in air using these unmanned aircraft systems that are flown in a location where we know Coccidioides to exist? And if so, what are the factors that are associated with airborne presence? And so we conducted a pilot study in the Carrizo Plain of California, which I'll talk more about in a little bit, but it is this area located in the Central Valley. So it's an area that's very endemic to Coccidioides. And we conducted our flights across about a 10 by 10 kilometer area. So we used two different sampling designs. First, we just flew the drones in ambient conditions about 10 meters off the ground, which was the distance where we thought that the rotor wash caused by the spinning drone blades was not going to aerosolize any of the soil. And then we also simulated these high dust events where we flew one drone low to the ground to kick up dust, and then another drone downwind in order to sample what was being aerosolized. So this is just another picture of that. Here's just a video. You can see the lower drone is kicking up some dust with the rotor wash, and then the drone is down. So overall, we did detect Coccidioides in about 5% of our air samples. Surprisingly, it was only in those ambient air samples that we found Coccidioides, which could just be because of the stochastic nature of detecting it and the challenges in like laboratory limit of detection mechanisms. But one thing that is interesting is that this is actually the highest height above the ground that Coccidioides has ever been detected in. So it's about 8 meters above the ground, and also the lowest ever volume of air filtered that detected Coccidioides. So we sampled about 160 liters of air. We didn't really see any relationship between positive samples and particulate matter, kind of expected given that we only had 2 positive samples. So what you're seeing here is just some of the distribution of particulate matter that we monitored using a purple air sensor on our drone. And then the vertical dashed lines are where the positive samples fell. We also looked at other factors like relative humidity, time of day, temperature. Didn't see much, again, not surprising given that we only had 2 positive samples. But we are planning to do, now that we've sort of validated this as an approach to monitoring Coccidioides in the air, we are hoping to do larger campaigns in order to tease out some of these relationships. So just to summarize some findings here, we from prior work have seen that ambient dust is associated with incidence. And so we're interested in looking at wildfires. In general, we see that wildfires are maybe not associated with incidents in California, but perhaps some of these larger fires located in areas where Coccidioides is associated. And we're really excited about these new methods of airborne spore detection that might help us parse out a little bit better how Coccidioides is being aerosolized and transported. Okay, so now I'm going to talk a little bit more about some other exciting fieldwork that we're doing on Coccidioides in California, and particularly how we're seeking to understand where Coccidioides lives in the soil. So up until this point, I really only asked you all to consider the environmental life cycle of the fungus and what is happening in the soil. But I've been hiding this whole other life cycle that Coccidioides has, which is the life cycle that happens within a host. So within a host, Coccidioides will initiate what's known as a parasitic life cycle. So instead of growing its mycelia, it'll transform to generate these spherules that can then replicate, spreading its spores again throughout the body. But we've been thinking about humans as the hosts, but what if the host is in fact not a human, but is a burrowing rodent that is living in the soil and also dying in the soil? Is it possible that the spores that are contained within this host body can be returned back to the soil? And so this is the basis for a emerging new hypothesis in the field that's called the small mammal endozoan hypothesis. And this postulates that rodent hosts are asymptomatically containing spores within their bodies in these granulomas. And then when they die, these granulomas dissolve, releasing the spores, and the rodent carcass is serving as an important source of nutrients for the fungus to develop its mycelia. And so this hypothesis is actually not a terribly new hypothesis, but it has experienced rise and falls over the years through a somewhat exciting, timeline. So back in the 1940s, this man here, Chester Emmons, was the first to say that rodents are known hosts for Coccidioides, and he was actually the first to say that rodents are the reservoir for Coccidioides. It is not soil. And then there was an outbreak in, I believe, 1957 near a Native American midden. And so researchers came in and they tested the soil for Coccidioides and they tested the rodents, and they only found Coccidioides in the soil. And so that somewhat squashed the idea that rodents are reservoirs for Coccidioides. There was a study where someone put a dead rodent that was infected with Cocci in the soil and showed that Cocci persisted in the soil for 10 years following that, but largely for the next several decades, the idea that Coccidioides was a soil saprophyte, lives in the soil, gets nutrients from the soil, prevailed, and all the studies doing environmental surveillance for Coccidioides tried to understand differences in soil characteristics. And what the studies found were two things. One, Coccidioides is really patchy in the environment and hard to find. Two, it's a lot easier to find if you sample rodent burrows. So that was kind of one bit of evidence bringing the rodent hypothesis back. And then second, there was a paper in early 2000s that did some comparative genomic analyses and found that the genes that break down animal keratin were upregulated in the family of fungi to which Coccidioides belong, and the genes that degrade plant cell walls were downregulated. So that led to the resurrection of the rodent hypothesis, which was published in 2019 by John Taylor and Richard Parker. So we wanted to ask, what is the role of burrowing mammals in shaping the distribution of pathogens within the environment? And what's been really challenging is that we have evidence that Coccidioides prefers to live in burrow environments, but to date, it's been near impossible to separate rodents from the burrows. And so we aren't able to ask, is it the rodents or is it the burrows? And we know that burrows have unique soil conditions. They filter water a little bit better. They maintain a nice temperature homeostasis. And so it's hard to say if Cocci likes burrows because of burrows or because of rodents. So this is where that site that I mentioned earlier, the Carrizo Plain, comes into play. The Carrizo Plain was designated as a national monument under the Clinton administration because it has the highest concentration of federally endangered wildlife in California. It is also California's largest native intact grassland. So it has a lot of really neat species, including foxes, badgers, mini rodents, etc. But the one rodent of particular interest is the one in the top left here, which is the giant kangaroo rat. And these giant kangaroo rats are considered ecosystem engineers because they create these precincts. So that's kind of shown on the right there, where they burrow in, and it creates these very distinctive mounds on the landscape. And these mounds you can actually see kind of from space because they clip the vegetation around those precincts. So in 2007, researchers wanted to study the effects of cattle grazing on these endangered rodents. And so they set up these little exclosures. So there are 20 experimental plots in the Carrizo Plain, and each one of them has this rodent exclosure in the middle where they've dug chicken wire fences 1 meter beneath the ground and trapped the rodents out, which leaves their burrows intact but abandoned for over 15 years. And so we sampled soil seasonally across a factorial design that crosses this rodent presence absence with burrow soil and surface soil. So each season we collected 500 samples. We started out going once every season, but summer was really hot and winter was really rainy. So we've since switched to a twice a year sampling scheme. So I'm showing results from 2 full years of sampling. So in total, as I mentioned, there's 20 plots. Each plot has this rodent exclosure in the middle, and we sampled the same sites each time, but in order for our first selection, we went to the plot, we divided the plot into a grid, a 7x7 grid, and then we randomly selected which grid we were going to go to. Outside the exclosure, we went to that grid cell, we located the nearest active looking precinct, and we collected 5 samples from the burrow entrances. We also collected surface soil in a transect that radiated outward from the burrow. And then within the exclosure, we did the same thing, but selecting precincts that looked inactive, which they all pretty much look inactive because these sites are actively. In addition, we collected information on some soil characteristics like soil moisture, soil temperature, vegetation level. Other groups on the monument are collecting information about rodent abundance. We did some soil infiltration rates. And then we conducted a mediation analysis with G computation in order to determine the total effect of rodents on Coccidioides, the indirect effect that operates via burrows, the direct effect that does not operate via burrows. So this would be due to maybe rodents either having the spore in their body and dying, or shedding their keratin into the soil, or potentially just changing the soil nutrients of like nitrogen through their feces or urine, etc. And then we determined the percent mediated. So in order to do this, we followed the approach from this 2015 paper by Wang and Ara, which has these sort of 3 steps here. So first, we modeled the mediator conditional on the exposure and covariates. Here, the mediator is burrows. In this case, you can't have a burrow without a rodent in real life. We could only have it because of our unique study design. So we actually just deterministically set this equation. The next step was to model the outcome. So Coccidioides presence dependent on the exposure rodents, the mediator burrows, and other covariants. So we built these models using generalized linear mixed models to account for our clustered sample design. And then you can simulate an intervention, so various levels of rodent presence and see what happens. Okay, so some of the results. So in the results I'm presenting here, we analyzed nearly 2,000 samples for Coccidioides, and we found that it was in about 16% of samples. If you look at the second bar there, those are the burrow samples with rodents. We see that about 30% of them have Coccidioides in them. Once you exclude rodents from the burrows, that positivity rate drops to about 19%. And then looking at the surface soil samples, we found very few of those had Coccidioides in them, but we do see a higher prevalence of Coccidioides presence in the surface soil samples where rodents are present compared to absent. So if we look at the results from our generalized linear mixed model, so these are the ones that are controlling for both burrows and rodents, we see that the odds of Coccidioides when rodents are present compared to absent is about 2.6 times higher than the odds of Coccidioides when rodents are absent. And the odds ratio for burrows is even higher. So odds of Coccidioides detection is 19 times higher in burrows compared to surface soils. The only soil characteristic that we measured that was associated with Coccidioides presence was soil moisture, which had an inverse association. And then when we put it into our mediation analysis, we see that overall, the odds of Coccidioides in soil is 32 times higher where rodents are present, but that about 73% of this association is mediated through burrows, meaning that about 25% is direct, which is a not negligible amount. We tried to add soil conditions to this to see if the different soil conditions in the burrows could be explaining some of this effect. And we see that it didn't explain too much effect. So we see that soil conditions maybe explain about 8% of the effect of burrows on Coccidioides because we did see lower soil moisture among them. I will say we measured just a very limited set of soil conditions here. We have partnered with Texas A&M, who are now doing a suite of a lot of more comprehensive soil characteristics, including total nitrogen and nitrates and other enzyme levels and a lot of very cool analyses. Okay, so just to summarize again, this section, so we've seen that Coccidioides is far more likely to be detected where rodents are present compared to absent. And this strong indirect effect suggests that burrows themselves are suitable microhabitats, even as rodents are likely amplifying the presence of Coccidioides. And as I mentioned, we're partnering with Texas A&M to further tease out what soil conditions might be important within these burrows. And I will note a key limitation here is that we're just using qPCR presence absence. And so we don't actually know if the fungus is viable, but we know that Coccidioides spores can hang around for a long time. And so maybe what we're detecting in these burrows or in the exclosures is just historical presence of Coccidioides. So I do want to talk a little bit about why does it matter if rodents are involved besides just like interest in understanding the role of rodents. One, I think it could have implications for public health protections. In California, construction workers receive education about valley fever, but they are the only occupational group that receives education. And other occupations are involved in digging in soil where rodents are present. If there's an endangered rodent, and construction is going to happen, people will come in and move the burrows, and those people have gotten valley fever. So I think it could have some nuances for trainings. I think it could help contextualize some of those climate influences that we talked about. So we talked about how valley fever is low during periods of drought and spikes afterwards. And that is very similar to what happens to rodent populations, which crash during drought and rebound sharply after. And if we think about other diseases that are associated with rodents, such as hantavirus, we see very similar patterns where hantavirus outbreaks tend to occur when rains return following a drought period. And then last, it could help us understand the geographic expansion of the fungus. So work by Morgan Goris and others has shown that the habitat suitable for Coccidioides is expected to expand across much of the Western US with climate change. And a key question is, will Coccidioides move to those new regions? And so prior study has found that both wind and small mammals are complementary dispersers of fungal pathogens. So spores can be carried in wind, but they can also be carried in rodent hosts. And so we need to kind of understand how rodent movement might be contributing to the spread of Coccidioides. And one additional idea that I am excited about, but not showing here, but have some funding to pursue, is thinking about how population susceptibility interacts with this process of geographic expansion. We know very little about how much of the population is already immune to valley fever. And so I think we need to dive more into population susceptibility and how that might interact with where Coccidioides is expected to spread over the coming decades. I'll just highlight this article, which was in the Washington Post about our work, and talked a lot about what I've presented today. And then last, I just wanted to spend a few minutes talking about some other fungal pathogens, because we are not in California here, but there are fungal pathogens that are present, including in Connecticut, and some of these are blastomyces and histoplasma species, which do have global distribution. Blastomycosis, I think, is a really fascinating fungal disease similar to Coccidioides. It lives in soil, although or decaying wood, but it does tend to prefer a little bit wetter environments. And as you can see, its distribution is across much of the eastern half of the US. And we see that the epidemiology of blastomycosis is changing. So the graph on the left is incidence rates from Minnesota, where we can see the sort of dramatic increase in blasto rates in dogs and humans. And then in Michigan this past year, there was the largest outbreak of blasto ever reported at a paper mill facility. So we're hoping to try to understand different environmental factors associated with these changes. Some hypotheses, again, is that it's this alternation between wet winters for fungal growth, but dry summers to allow the fungus to spore. In parts of the US that have usually been covered by snow in the winter, we think some of these rain on snow events could be allowing the fungus to spread in the winter months. And CDC does report more outbreaks of blasto in winter months, whereas previously they did not see that. And then we're also interested in flooding, which could transport the fungus to new areas. And then histoplasmosis is another disease which is across much of the eastern US. And I think what is really interesting there is its connection with bats, because most of the outbreaks of histo are associated with the guano of birds or bats, but it's not really known if bats are actively carrying the disease themselves, or if it's just their feces that are creating the right soil conditions for histo to grow. So with that, I will again acknowledge my collaborators, and I'd be happy to answer any questions.
You looked at if the El Nino or La Nina impacts this?
Yes, so I have not personally, but Alexandra Heaney, who's a new assistant professor at UC San Diego, has started looking into that. And yes, I mean, it seems to align with the phenomenon of wetter winters is allowing Coccidioides to grow, and the drier winters is not allowing that process to happen. So we do see pretty strong effects with El Nino. And I think what is interesting there is because El Nino and La Nina events are a little bit more predictable. We're starting to think about whether El Nino or La Nina events can be used as an important forecasting tool for valley fever, just to give us a little bit more.
I know very little about fungal infections, but I was just curious, like, is there anything known about like if there's a safe exposure to like fungal spores, or it's just like any exposure like put you at risk?
Yeah, that's a good question. So about 60% of people do not have symptoms for valley fever. And so it's possible that those people had lower doses of spores. It's not actually known the infectious dose. People have speculated 1 spore is enough to cause disease. I tend to think it's a little bit higher than 1 spore, but I do think it's probably pretty small, just because detection efforts in air usually return null results, which I think suggests it is related to lower limit of detection in the lab. Usually the laboratory methods need about 10 spores to detect. So if we're not filtering that much, yeah, so I guess I'll say no, but probably small. But some of the people with very severe disease, we speculate it's because they have a big dose, likely because they were like in yeah.
Kind of building off of that, because there's so much subclinical disease with cocci, and then also more, and there's such a wide spectrum of disease with immune compromised individuals having disseminated disease and meningoencephalitis. Did you look at all if your case definition changed at all, like if you had more reporting of subclinical disease, or if you had more immune compromised people, because there might be more immune compromised people in the population with people getting older and more diabetes that are affecting your results in any way?
Right, yeah, that's a really good question. We have very little information from the surveillance record on what symptoms the person in the record has. This past year, California Department of Public Health did an enhanced surveillance study where they called people who tested positive for cocci, and about 86% of the people who were reported did have symptoms, but that other 14% did get picked up because they were maybe about to undergo a kidney transplant and they needed to know if they had a fungal infection for that. So there is certainly some picking up of cases that are flying under the radar in the surveillance. I have not looked personally at associations between like rise of immune compromised populations and the rise of cocci. I think it would be really interesting to do so, and also important to tease out, you know, how much of these changes in incidence rates is due to environmental factors versus changes in the
I think we could probably take one more quick question.
Yeah,
Sure. So my bit curious, why did you use soil moisture as a potential predictor instead of rainfall, looking at the life cycle of valley fever?
yeah, so you mean in the field study?
No, I'm talking about the model.
Oh, the model,
Yeah, instead of
so
using rainfall, why didn't you use soil moisture, looking at the life cycle?
yes, that's a good question. This might be a bit of an unsatisfying answer. I considered soil moisture, but soil moisture, at least what I was using is a modeled product. And the story wasn't as clear, I think, because the product that I was using had a lot of variability in its modeled estimates of soil moisture. And I've since learned some new products that maybe would have been better to use. But I also was curious about how to tease out the relative influences of precipitation and temperature, because very different phenomenon are happening with respect to precipitation and temperature in California and other parts of the world. So temperature, we're kind of seeing steady increases in temperature over time, whereas precipitation, we're not necessarily seeing that the West is going to get wetter with climate change, but we're seeing that precipitation is going to be more concentrated in specific times of year. So I do think it would be interesting to repeat this using soil moisture, but I also just kind of wanted to build intuition using the sort of fundamental components of soil moisture first.
Thank you so much.
Guess it's find a way to